Good morning. Thank you for joining me today. I'm going to be going over with you CAR T-cell at UCLA, engineered cell therapy progress being driven at UCLA. Just to remind you, you can ask questions on Twitter and Facebook, and I will address them at the end. To give you the outline for today's talk, I'm first going to go over the rationale for chimeric antigen receptors, CAR T-cells. That background will then provide you with the information needed to understand the FDA approvals of the 2 approved CAR T-cell therapies that are being used here at UCLA, to go through the clinical trials at UCLA, and also to review the preclinical development, so what we're getting ready to put into patients in order to improve the safety and the efficacy of this very exciting therapy. So of course we need to put this into context of the timeline of immunotherapy. We've been working on immunotherapy for a very long time. Allogeneic stem cell transplant could be considered the ultimate immune therapy because we're giving patients a completely new immune system. So we take the stem cells from one person and then give them to a patient. That provides patients with a new immune system that is not specific. So although it's an all-new immune system, it does not have the specificity that we need. A lack of specificity increases toxicity, which is why we're continuing to try to improve our immune therapies to make them safer and more effective. After allogeneic stem cell transplant, another I like to mention is rituximab. This is an anti-CD20 monoclonal antibody. Anytime we talk about monoclonal antibodies, we describe it as anti and then the target. So what is that antibody tagging? Tagging for the immune system to attack. CD20 is one of the markers on the surface of B cells. B cells are one of the cells that become malignant or cancerous in lymphoma. So an anti-CD20 monoclonal antibody, rituximab, targets B-cell lymphomas in order for the immune system to attack them. Now, just having the antibody is usually not enough for patients with blood cancers. The reason for that is because there's immune dysfunction. So in leukemia, lymphoma, multiple myeloma, the immune system is not normal. So while we can tag the lymphoma with the antibody, the immune system may not be able to fully take care of it. The next generation I want to point out to you on this slide is blinatumomab. This is a bispecific antibody, meaning that it targets a cell surface marker on a leukemia, but also brings the T cells into the vicinity in order to help improve and overcome that immune dysfunction that's there. But just having that one extra marker, which on this is CD3, to bring the T cells into the vicinity is usually not enough with the amount of immune system abnormalities that can be present in leukemia, lymphoma, and multiple myeloma. So in order to get around that, we bring together the antibody therapy and T-cell therapy that is the engineered cell therapy of CAR T-cells. So let's look at the normal cells of the immune system to find out how we're harnessing B-cell and T-cell functions in order to create CAR T-cell therapy. We do this by using what is considered the variable region. You'll see this on the CAR T-cells as a single chain variable fragment. Whenever we're looking at these cartoon diagrams of the immune system, this is going to be outside the cell. This is going to be the lining of the cell. We call it the cell membrane. And inside of the cell will be down here. Same thing with the T cell. This is outside the cell. This is the cell lining, the cell membrane, and then inside of the cell. So B cells, this is where we're going to have the tagging, the identification portion. Then the T cells, that's where the cell killing capability can come through, where inside we have that CD3 that I already mentioned. Now, T cells need more than just CD3. They need an extra, what we call co-stimulatory molecule in order for them to be activated. This is an important regulatory part of the immune system to prevent autoimmune diseases. So T cells aren't just activated all the time, they're only activated in certain instances. The reason that we have that, again, is to prevent autoimmunity. One of those co-stimulatories is CD28. So taking what we know about B cells and T cells and their function, we can put it together into a chimeric antigen receptor. Again, looking at the signaling, the tagging part of the B cell on the outside of the cell of a CAR T cell. Then there's the connection to the inside, which will have the T cell machinery, meaning that without the patient's immune system being functional at all, you can tag the tumor cell and have activation of the T cell to fully eradicate the cancer. So just another depiction of this so you can understand what we're talking about as we go through the clinical trials. This is a single chain variable fragment. Now, in the majority of the clinical trials that I'm going to be going through, that marker is CD19. So just like with antibodies, when we talk about what CAR T cells target, it's anti-CD19. That's not the only target, but for the CAR T cells I'm talking about today, it's anti-CD19, meaning this outside portion, the single chain variable fragment, the antibody portion, is targeting CD19 because it's on leukemia and lymphoma. Once it binds, it then targets to the T cell machinery to start killing those leukemia or lymphoma cells. These have been in development for some time. That's why there's first, second, and third generation chimeric antigen receptors. So the assembly line. So part of why I included how we make these is because about 5 years ago, when chimeric antigen receptors were first coming into the news, there was a headline that said, HIV cures girls' leukemia. So of course you can imagine with that, I was receiving calls from all of my family members and friends about how on earth are we using HIV to cure leukemia. The reason for that is that we use an inactivated HIV virus in order to package the CAR T cells and have them be expressed on normal T cells. So the way we do this is we take the genetic code for a CAR T cell. So this is going to be the genetic code that says CAR. We then put it into a packaging cell line along with the components of the inactivated HIV virus. So that HIV virus can't replicate. All that it can do is package the CAR into what we need in order for it to be delivered into the T cells. So once we have that packaged, we then activate a patient's T cells, put it together with either our retroviral particles, you'll sometimes hear that referred to as a vector. By doing that, we then make T cells that express CAR on them. So it's an amazing process in how we're using inactivated HIV in order to cure leukemia and lymphoma. This is what the patient process looks like. So we first screen patients. So although this is an incredibly effective therapy, just like all other therapies, there are side effects. So we need to make sure we choose our patients appropriately, and we do that through our own screening process. Once a patient has passed the screening process, we then collect their cells. This is called apheresis. Apheresis essentially looks like a dialysis machine where the blood is taken out of the patient, we take their T cells, and then the blood goes back in. We then take those T cells, modify them so that those T cells now have CAR on them, and then bring them back to UCLA. Once we have the cells and we know they've passed all of their quality controls, we then give patients small doses of chemotherapy. This helps to increase how well the CAR T cells can work and make sure that the patient's immune system doesn't dampen the CAR T cell response. We then infuse the CAR T cells just like a blood transfusion. They go into the patient's vein, then they find their way to all the leukemia or lymphoma cells all throughout the body, and then we monitor for any side effects. So these are the 2 FDA-approved therapies. The first one is for acute lymphoblastic leukemia. You'll see that abbreviated ALL. The second is for diffuse large B-cell lymphoma, abbreviated DLBCL. Now this is Axicell. The trade name is Yescarta. The ALL, they have not yet given us a nice short name like Axicell, so we can just call it Tisagen, but Kymriah is the trade name. So this is why chimeric antigen receptors were needed in ALL. This shows the outcome of patients who were treated for ALL and either their leukemia didn't respond or it came back after they had a response. So what you can see here is, unfortunately, the overall survival rate is 7% for patients who, if their leukemia either didn't respond or it came back. That's an incredibly low survival rate and shows the unmet clinical need for patients with ALL before we had CAR T cell therapy. This graph shows survival on the y-axis and time on the x-axis. The different lines show different patient age groups. So the younger patients have about 12% at 5 years, where patients that are greater than 50 years old have only 3% at 5 years. Now keep this in the back of your mind as I go through the data for the CAR T cell therapy in ALL. So here are the 3 major trials that have looked at treating ALL patients with the anti-CD19 CAR T cells. What we can see is complete remission rates, so eradication of the leukemia, in 73 to 93% of patients, both in pediatric and adult patients, and we've treated more than 100 patients. So now look at this survival curve. You can see the major difference between what I showed you before and the survival curve with CAR T therapy for ALL. So again, survival on the y-axis, months on the x-axis. So in contrast to what we saw before, where most patients were dying by 1 year, 79% are still alive when we use CAR T-cell therapy, an incredible breakthrough for these patients. Similar situation for diffuse large B-cell lymphoma. So in diffuse large B-cell lymphoma, patients will receive upfront therapy. If they go into a CR, which is a complete remission, they are sometimes cured. But in patients who either do not respond to that first therapy or the lymphoma comes back after they've been treated, the complete remission rate is only 8%, with a median survival of only 6 months. So you can see that we lose patients really within the first 12 months if they don't respond to upfront therapy. Same thing, the 3 trials that have been presented for the treatment of diffuse large B-cell lymphoma with anti-CD19 chimeric antigen receptors. We're seeing complete remission rates from 43 to 58%. Contrast that to 8%. That's how big of a difference this is. And then patient populations, this is diffuse large B-cell lymphoma. The other abbreviations there, so TFL is transformed follicular lymphoma. So that's a diffuse large B-cell lymphoma where there was a low-grade lymphoma first and then it turned into a diffuse large B-cell lymphoma. And primary mediastinal B-cell lymphoma, which is a subtype of diffuse large B-cell lymphoma. So across the spectrum of diffuse large B-cell lymphoma that either didn't respond or relapsed, we're seeing very favorable response rates. At the bottom, we have a PET-CT scan. So just to review a PET-CT scan, this is at the level of the pelvic bones. On a CT scan, bone is white, air is black, and gray is soft tissue, everything in between. So we essentially take 5mm cuts throughout the patient in order to get these pictures to go through the CT scan. The PET portion is the metabolic activity, the lymphoma activity. So you'll hear this talked about as PET positive, FDG avid, or hot are the ways we sometimes describe a PET CT scan. So here you can see the patient at baseline had this large area of lymphoma, but just 30 days after treatment had complete resolution with no PET activity seen. So again, just showing visually the power of this anti-CD19 CAR T-cell therapy. So a major leap forward in diffuse large B-cell lymphoma. So just like in ALL, you look at the curves that were seen before CAR T-cell therapy and then afterwards. So 52% are alive at a median of 15.4 months follow-up. The median survival I showed you before was 6 months. So this is a more than doubling of overall survival for these patients. And of course, this is what led to the FDA approval here of Axi-cel. This is in adult patients with relapsed or refractory diffuse large B-cell lymphoma after 2 or more prior lines of therapy or acute lymphoblastic leukemia. Now this is only for patients up to age 25. So age 25 and younger is for acute lymphoblastic leukemia. That's either relapsed after 2 lines of therapy or didn't respond upfront to therapy. So of course, this is excellent response rates. We're seeing improved survivals, all very exciting, but we know we can't just rest with this and we need to continue to improve. So this is the clinical trial portfolio that's available at UCLA right now. The first is talking about multiple myeloma. So multiple myeloma is like a sister disease to lymphoma. It's a B cell, but as B cells continue to mature, the last stage of maturation of a B cell is a plasma cell. Now when B cells go through that last stage of maturation, they change what's on the surface of their cells. So CD19, which is present on lymphomas, is no longer present on plasma cells, so it's no longer present on multiple myeloma. So the target that we use, that single chain variable fragment for multiple myeloma, is B cell maturation antigen, BCMA. Now we have this study available, the KITE 585-501. The other trial that's been presented, that's not enrolling right now, had a response rate of 80%. Now to put that into context, the last FDA-approved therapy for patients with greater than or equal to 3 prior lines of therapy had a response rate of 30%, and we were incredibly excited about it. So most recent FDA approval in this patient population, 30%. Most recent CAR T-cell trial reported response rate of 80%. So really promising therapies in multiple myeloma. This is a disease where we've had incredible progress, but it's still treatable but not curable, and that's what we're working on here at UCLA. For mantle cell lymphoma, this is the ZUMA-2 trial. This is again an anti-CD19 CAR. Because this is a lymphoma, it still has CD19. So in patients with mantle cell lymphoma who are relapsed or refractory, they could go on this anti-CD19 CAR T-cell therapy. We also have ZUMA-5 for anti-CD19 CAR T-cells. That's relapsed or refractory lymphoma that's low grade. This is primarily follicular lymphoma. I had already mentioned that because that's one of the low-grade lymphomas that can transform to diffuse large B-cell lymphoma. But now, even before it's transformed, we could consider giving CAR T-cells to patients. Lastly, at the bottom, you'll see diffuse large B-cell lymphoma and the Belinda trial. Now, we know that CAR T-cells already approved for diffuse large B-cell lymphoma after 2 prior lines of therapy. But those promising results have caused us to ask, should we bring this earlier in therapy? So right now, patients, if they had diffuse large B-cell lymphoma that either didn't respond to therapy or relapsed after the therapy, we would have them go on to salvage chemotherapy and an autologous stem cell transplant. Because of how promising the CAR T-cell results are, the Belinda trial is going to take patients and randomize them either to the standard therapy with salvage chemotherapy and an autologous stem cell transplant, or to go on to the CAR T-cell therapy. One of the very exciting things about this trial is that patients that are in the standard therapy arm have the ability to cross over to the CAR T-cell arm. So if they don't respond to salvage chemotherapy, they can automatically go on to get the CAR T-cell therapy. And we don't just have these industry-sponsored trials here at UCLA, we're also doing our own research to look at how can we improve our target selection to hopefully overcome resistance. How can we use this therapy in cells, not just T cells, but maybe other cells of the immune system and have a more renewable source of cell-engineered therapy? And then also to see how can we have safety modifications. So when we talk about targets, it's important to talk about how do we choose our CAR T-cell targets. So what we want to do is find a target that is present on tumor cells, but not on normal cells. Now, I've been talking about CD19 this whole time, and CD19 is present on B cells. So if you go to the graph on the bottom here, this is how B cells mature. They start in the bone marrow, they leave the bone marrow to go to the peripheral blood, the blood that's circulating in the veins, and at the end stage of their maturity, they become a plasma cell, like I mentioned. So this is the cell that becomes cancerous in multiple myeloma. So all of these cells, when they become cancerous, still have CD19. But CD19 is also present on B cells. We know from our extensive experience with rituximab that patients can survive if we monitor and manage them correctly without B cells there. That's why CD19 is such an excellent target, because it's present on a range of B cells, so it's present on a range of cancers, and we know it's a safe target, meaning if we take out those cells, there's not toxicity there. We also want to make sure that it's persistent. So some markers on the cell surface come and go, but we want to make sure we're finding one that stays there so we can have a persistent therapy. We also want there to be high affinity, meaning that the CAR T cell, that single chain variable fragment, can bind well to it and that it won't just let go, because of course in order for the CAR T cell to work, it needs to be bound to the tumor cell. So when we look at what other potential targets could be there, CD20 and CD22 are also in development right now, and that's because they're also present on a range of B cells. It's one area of specific research here with myself and Dr. Yvonne Chen about how can we overcome the resistance of CAR T cells. So we know that they're very effective, but patients do relapse afterwards and they can become resistant. One of the ways the tumors get around CAR T cell therapy is by downregulating or getting rid of CD19. It's called antigen escape. So the tumor cell gets rid of CD19 or the CAR T cell target. That's what we call antigen escape. If we look at the previous trials, the rate of antigen escape is anywhere from 30 to 100% depending upon the trial that you're looking at. So what we're doing here is engineering a CAR that targets more than one of the cell surface markers, what you call a bispecific CAR. So going back to the diagrams that we looked at before, so this is outside the cell, this is inside the cell. The single chain variable fragments, the areas that are going to target the tumor, instead of having just one, you have two there. And what we're working on is CD19 and CD20 together. So Dr. Chen and I are developing the protocol for patients with diffuse large B-cell lymphoma and other non-Hodgkin's lymphoma that have relapsed in order to have a CD19 and CD20 CAR in order to decrease antigen escape and CAR T-cell resistance. One of the other things that we're doing is looking at the cell type. So again, what I showed you in that data of the FDA-approved therapies is chimeric antigen receptors in T cells. But we can also put chimeric antigen receptors into other cells. One cell that we can do is stem cells. So hematopoietic stem cells are the cells that are located in the bone marrow and give rise to all of the blood cells. By putting a chimeric antigen receptor into a stem cell, we then create a renewable, multi-lineage chimeric antigen receptor therapy that will be persistent. So we won't lose a response by losing the T cells because the bone marrow will continue to produce an anti-CD19, anti-BCMA therapy. We can do this with TCRs, which are T cell receptors, which are very similar to CARs, as well as CARs. We have extensive experience with this at UCLA. So Dr. Cohn's laboratory is actually the first to treat sickle cell disease by modification of stem cells. And we've also had treatment of severe combined immunodeficiency by using not a CAR, but gene modification therapy in order to cure these diseases. We can also have modification of select T cell subsets. That's the other thing that we're looking at in the protocol with Dr. Chen is if we use not just all T cells, but selected T cells, are we going to have a safer approach to CAR T cell therapy? This is just an example of NY-ESO as a target. So we've talked about BCMA, CD19. NY-ESO is another target for multiple myeloma, as well as some of the solid tumors, including melanoma and sarcoma. With this clinical trial that was developed here at UCLA, we modify both stem cells and T cells. So we say on day 0, the patients received their modified stem cells. The next day, they received the modified T cells. Those modified T cells are the ones that go in and attack any myeloma cells that are there currently. But again, we worry about persistence with T cell modification. That's why we have the modification of the stem cells, because as those cells continue to grow, they'll have a renewable source of anti-myeloma therapy for these patients. So again, the example here at UCLA, how we're modifying both stem cells and T cells with engineered cell therapy. Lastly, we want to have improved safety, and part of how we do that is with a suicide gene or an off switch. So in that genetic material of the CAR, we can put in an off switch. What that means is that there will be a drug, GCV, which stands for ganciclovir, is the most commonly used, where by giving that drug, we can completely turn off the CAR if the gene is present in those cells. We've done that here in the lab, and that's part of the off switch in the NY-ESO TCR. But there are other that are in development here, including EGFR, CAS, and multiple others. All of these working to be a potential off switch to turn off the therapy in case there's undue toxicity or for any reason we decide that it's not safe for the patients to have that therapy anymore. So in conclusion, engineered cell therapy brings new hope to patients that did not previously have any therapeutic options. Here at UCLA, we have a comprehensive engineered cell therapy clinical trial portfolio, and there's exciting new research to bring safety and efficacy to this already very promising approach. I just want to thank my colleagues, both the adult and pediatric colleagues who treat these patients and do research with me. In particular, I want to highlight Josh Sassine, who's the clinical program director for our CAR T-cell program, Dr. Gary Schiller, who is our stem cell transplant director, and on the pediatric side, Dr. Donald Cohn, who's one of the pioneers in engineered cell therapy and was my laboratory mentor. I also want to bring up the partnership between UCLA Health and Leukemia and Lymphoma Society. Leukemia and Lymphoma Society is an incredible organization for patients and their family members and very supportive of research. They're partnering up with us on our Closer to a Cure, our campaign to continue to promote CAR T cells in order to bring it to more patients to have these promising outcomes. We're having an event on April 10th that everyone is welcome to join us at Santa Monica Hospital. And just to bring in my own partnership with Leukemia and Lymphoma Society, one of my patients, Lila Javon, and I started a team and training event with Leukemia and Lymphoma Society called Climb to Cure. So this is a picture of us at the top of Mount Kilimanjaro at the inaugural hike in January of 2017, and just another picture of the mountain. With this, we raised $1 million for Leukemia and Lymphoma Society within 1 year of starting it. So I want to thank you for your attention. It's been wonderful having you join me today, and I'll take any questions on Twitter or Facebook. Thank you. Okay. The first question is risk versus benefit of CAR T-cell therapy. Is it lower risk to chemotherapy or other modalities? This is an excellent question. CAR T cells have significant toxicities associated with them. Because of our experience on the clinical trials, we know how to mitigate those toxicities, but they are very real. Now, other modalities that have similar toxicity profiles would include stem cell transplant. Although it's early in the treatment, it does appear that these are likely safer than an allogeneic stem cell transplant. There are so many treatment options, it's difficult to say how it matches up with each of the different chemotherapy options, but in general, these are toxicities that we know how to treat and can address safely for patients. The next is, when is an absolutely obvious time to use CAR T cell therapy? It's at the time where there are no standard options, so standard of care options, or clinical trial options available to treat patients. So using the example of multiple myeloma, there are multiple approved FDA therapies and clinical trials currently in progress. But part of why we're so excited about the anti-BCMA CAR T cell trials is because these are patients that do not have any other lines of therapy available, either through clinical trials or standard of care. The last is, does CAR T cell work on other types of cancer other than lymphoma? All very good questions. So yes, it does. So I mentioned that it works in leukemia and multiple myeloma. CAR T cells have been slower to be developed in the solid tumor oncology space, meaning things like breast cancer, lung cancer, GI cancer, but there are other therapies that are being developed with engineered cell therapy for those cancers. The example that I gave is with NY-ESO. So that's a T cell receptor, which is similar to CAR T cell. It's an engineered cell therapy that's being used for melanoma and sarcoma that's been developed here at UCLA by Dr. Tony Rivas. Any other questions? All right, thank you for joining me.